Assalamualaikum and a very good morning to Dr. Balqis. My name is Izin Ashoha and my group is Nur Shamimi, Anisha Ira, Siti Nura Halifa, Muhammad Tajuddin and Nurul Adlin are going to present our practical 6, which is the application of cellulase and hemicellulase via extraction of juice from fruit.
Hi, I am Hanifa. In this video, our group will present about our experiment, which is application of cellulase and hemicellulase in extraction of juice from fruit. In this experiment, we want to observe the effect of cellulase and hemicellulase on juice extraction. Basically, we use two types of enzymes in this experiment, which are cellulase and hemicellulase. And the reason why we use this enzyme is because this enzyme will help in producing higher yields and clarity of the juice extraction. Furthermore, the use of maturation enzyme also increase the extraction yield and improve processing without extra cost. Apart from the types of enzymes that we use in this experiment, they also have several factors that can affect the yield of apple juice, such as incubation time, which are 40 and 80 minutes, temperature for incubation, which are 50, 55, and 60 degrees Celsius, and lastly is the pH condition. In terms of the procedure, we must do in detail to avoid any failure, such as chopping to increase the surface area, carry out control, and filtering. For the result that we obtain in this experiment, the higher yield of juice can be calculated by using the formula of enzyme minus with the control. So, at the end of this experiment, enzyme activity of cellulase and hemicellulase in producing a higher yield of juice can be compared under optimum pH, temperature, and incubation time. Fruits are important sources of essential dietary nutrients such as vitamins, minerals, and fiber. Fresh fruit are highly perishable due to their moisture content of more than 80%. To extend the shelf life, it is necessary to process the fruit by converting it into products like juices, fruit bars, and many others. Fruit juice is the product for direct consumption with the largest quantity of processed mature fruits. Basically, we can obtain the juice from the fruits through the extraction process. Various types of enzymes are involved in juice extraction. So, the purpose of the enzyme is to improve the yield, enhance the clarity and stability of the juice by degrading soluble pectins and starches that would otherwise cause haze. Generally, most of the fruits contain a high quantity of pectin, making the extraction difficult by conventional methods. In this experiment, there are two types of enzymes involved in extraction process, which are cellulase and hemicellulase. So, the cellulase and hemicellulase play a significant role in the conversion of cellulose and hemicellulose to sugars.
For the apparatus that is needed for the experiment is knife, small filter tunnel, clustering rods, spatula, 100 ml beaker, 250 ml beaker, aluminium foil, water bath which consists of 50, 55 and 60 degrees Celsius, 10 ml or 25 ml measuring cylinder, 1 ml pipette and tip, and stopwatch. For the chemical reagent that is needed for this experiment is green apple, enzyme, distilled water, and coffee filter paper. For the procedure, the green apple is chopped into smaller pieces by measurement of 5 mm times 5 mm times 5 mm. Then this green apple, the cube green apple is weighed and transfer 40 gram of the apple into a beaker. The beaker then covered by aluminium foil and then incubated in a water bath at 50 degrees Celsius for 40 minutes. And then The experiment is carried out as described above using heat and activated enzyme by boiling for 15 minutes. After that, the apple juice is filtered from the apple pulp using coffee filter papers and funnels. The last one is the volume of the juice obtained is recorded by 30-second interval up to 3 minutes, 3 minutes up to 10 minutes with 1 minute interval between it, until all the juice has separated from the pulp. The effect of enzyme reaction is by doing the incubation. So the activated and inactivated cellulase is incubated at different time and different temperature, which is 40, 80 minutes, and the temperature is 50 and 55 degrees Celsius. Each of the activated and inactivated cellulase has 4 beakers to incubate at different time and different temperature. And then the incubation is repeated with hemicellulase enzyme.
So back to the result. So from we have table 1 and table 2. For table 1 is juice yield ml for sample treated with cellulase and heat inactivated cellulase control for different incubation time at incubation temperature of 50 degrees Celsius. So we have the incubation time, which is 40 and 80 minutes, the time interval of measurement in second, 30 to 600 second, and the juice yield is cellulase and control. And for table 2 is juice yield ml for sample treated with hemicellulase and heat inactivated hemicellulase on control for different incubation time at incubation temperature of 50 degrees Celsius, which is the same as table 1. Incubation time is 40 and 80, time interval 30 to 600 second, and the juice yield is hemicellulose and control. So we convert the data into graph 1 and graph 2. Graph 1 is a graph juice yield for sample treated with cellulase and heat inactivated cellulase for different incubation time 40 minutes at incubation temperature of 50 degrees Celsius. The left one is for 40 incubation time 40 minutes. The right one is for 80 minutes. And for graph 2 is hemicellulase for 40 minutes and also 80 minutes. Next is for table 3 and table 4. So table 3 is the same as table 1 and 2, but this is for the incubation temperature 55 degrees Celsius, which is the left one is for cellulase and the right one is for hemicellulase. So we convert the data into graph 3 and graph 4. So the graph 3 is for cellulase and graph 4 is for hemicellulase. So the left one is for incubation time 40 minutes and the right one is for incubation time 80 minutes.
Assalamualaikum Dr. Balqis. I am Nur Shamimi binti Abu Jalil. So now I will proceed with the result part, and this slide is show the table 5, volume of the juice for the sample treated with the cellulase and heat inactivated cellulase for different incubation time. While table C is the volume of the juice for the sample treated with hemicellulase and heat inactivated hemicellulase as the control for different incubation time. Both enzymes are carried out at the same incubation temperature of 50 degrees Celsius. Based on the table that has been calculated, then graph 5 and 6, which are graph volume of the juice for a sample treated with different enzyme for a different incubation time at the incubation temperature of 50 degrees Celsius are constructed. Then here is the table 7 and table 8. The difference is those enzymes are run under incubation temperature of 55 degrees Celsius. This slide represented the graph 7 and 8, volume of the juice for the sample treated with different enzymes which are cellulase and hemicellulase for different incubation time at incubation temperature of 55 degrees Celsius. In order to see the clear comparison between those two enzymes, then we decide to make graph 9 and 10 where they show the pattern of the enzyme. Here is the calculation part where to obtain the volume of the juice. By then, we need to use the formula enzyme minus with the control. For enzyme cellulase, then the formula will be cellulase minus with the control. This is one of the example calculation for the volume of the juice of the sample treated with the cellulase and heat inactivated cellulase for a different incubation time at the 50 degrees Celsius is shown. The same calculation was performed for the both enzymes, which are cellulase and hemicellulase, from the time interval of 30 to 600 for different incubation time in second at different incubation temperature.
So now I will proceed for the discussion part. For this experiment, the first thing that we should do, as the apple fruit in the solid state, we have to cut off the apple into small pieces to increase the surface area of the fruit. This to ensure that the efficiency of the solid liquid extraction at the higher yield, and the reaction will be easier by the presence of the both enzymes, hemicellulase and the cellulase. So this is the example on how the fruit or any raw material has been cut off in the cube shape. There are several factors which can affect the yield of the fruit juice, which are incubation time, type of enzymes, temperature at the ambient temperature or incubation temperature, and the pH condition. For this experiment, the incubation time, there will be the 40 and 80 minutes. The type of the enzymes that will be used to speed up the reaction of the extraction are hemicellulase and the cellulase. The temperature for incubation, which are 50 and 55 degrees Celsius, while the ambient temperature, which is the atmospheric temperature in the laboratory, which is for about 20 to 22 degrees Celsius, and the pH condition of the fruit and the juice, which are the acidic or basic.
Generally, we obtain higher yield of apple juice extraction when treated with enzyme. Based on the graph, the volume of apple juice is higher at the incubation time of 80 minutes when treated with cellulase or hemicellulase for both temperature at 50 degrees Celsius and 55 degrees Celsius. The highest volume of sample when treated with cellulase at the incubation time of 18 minutes based on the graph 10. Next part is the effects of different enzyme and incubation time on juice yield. The volume of apple juice yield is higher when treated with hemicellulase than cellulase. This is because hemicellulase can easily hydrolyze the hemicellulose, as hemicellulose is hemophilous and short branch structure, while the cellulase is the long unbranched structure and resistant to enzymatic degradation of cellulase. Next, the incubation time of 18 minutes produced the higher yield of juice compared to the 40 minutes of incubation time. This is because longer incubation time can hydrolyze more cellulose and hemicellulose.
The other effect that will influence the yield of the fruit juice, which are the pH condition of the enzyme itself and the temperature. For the pH, we have to make sure what kind of the pH condition for the both enzymes and the raw materials itself to ensure that the catalyst mechanism of the enzyme substrate will be easier and strong enough to build the linkage between the enzyme substrate complex. This is because the enzymes are very specific. The hemicellulase and the cellulase are two different enzymes which have the different components of the nucleophilic and the electrophilic. They either have the high positive hydrogen ion or hydroxide ions. The optimum pH for the hemicellulase is at 7, which is the condition will be neutral. But for the higher enzyme activity of the hemicellulase, there will be 5 to 9 of the pH value, while for the cellulase there will be the 6.5, which is the weak acidic condition required for the enzyme activity. The next one is the temperature. The temperature also one of the major factors which we have to concern for this experiment. This is because different temperature will have the different yield of the production for the fruit juice or other enzyme activity. The high temperature, either the incubation temperature or the ambient temperature, will tend to have the high yield of the product, but the too high of the temperature will lead to the denaturation of the enzyme structure, and the yield of the juice will be lower. So for the optimum temperature for this experiment, there will be the 50 to 55 degrees Celsius for both enzymes, which is the 55 degrees Celsius is the highest temperature for producing the high yield of the fruit juice. The ambient temperature also we have to make sure, we have to concern, because the ambient temperature can be the temperature that will influence the extraction process, and the ambient temperature will be the room temperature of the lab, maybe for about 20 to 22 degrees Celsius. So now I will proceed for how enzyme works in increasing the yield of the fruit juice. Based on the figure 1, it shows the extraction process of the fruit, which is the apple, in the solid to the liquid components by using the enzymes. The hemicellulase and the cellulase will break down the structure of the fruit cell and the cell wall of the fruits and release the some reducing sugars. Those enzymes will be hydrolyzed enzymatically to increase the yield efficiency of the fruit juice. But there are some problems, which are low yield of the juice, sedimentation occur, and the juice produced become cloudy. Therefore, to reduce the sedimentation, which is the sedimentation is actually the two different phase layers formed. So to avoid that, we have to put the additive that should separate the solid structure of the fruit, or we have to use the correct and new plastic funnel and the filter paper to ensure that the fruit solid structure will be extracted greatly and correctly. So for cloudy condition, we also can add the pectinase to break down the pectin or any excess polysaccharides that might occur, that might influence the solution to become cloudy. So this is the example of the degradation of the cellulose, which is the complex sugar component in the fruit by the cellulase enzyme. The cellulose will be in crystalline and amorphous region. As we know, mostly the cellulose will be positioned partly in the crystalline region, which is quite difficult for the degradation of the enzyme to occur, and the bond among them will be quite strong to be broken. That's why the hemicellulose in this experiment have the high yield rather than the cellulase, due to their difficulty based on their sugar arrangement in the cell, and the hemicellulose can be easily hydrolyzed rather than the cellulose.
In the juice extraction, there are two enzymes that also can be used, which are pectinase and amylase. Pectinase is used for the clarifications of the juice by breaking the polysaccharide pectin structure present in the cell wall of plants into galacturonic acid monomers. The pectin structure breakage facilitates the filtration process, and it increases the total yield of juice, while for the amylase role is to degrade the starch with the pectinase. It also eliminates the possible actions of starch molecule agglomerating with proteins, pectins, and thus eliminate haze formations, and also eliminate the possible cloudy juice formation. In this experiment, the highest yield of apple juice is at the incubation time of 18 minutes treated with the hemicellulase enzyme, and at the temperature of 55 degrees Celsius.
As a conclusion, the volume of juice yield is higher when treated with the hemicellulase than cellulase. The incubation time of 80 minutes is produced the higher yield of juice compared to the 40 minutes of incubation time. The highest volume of apple juice is at the incubation time of 80 minutes, treated with hemicellulase enzyme at a temperature of 55 degrees Celsius.
This is all our references, and that's all for our presentation. Thank you so much, doctor, and take care.